Hi everybody, this is Mrs. Kraus, and this is our Unit 2, Part 4 video on enzymes. In biology class, we often study chemical reactions because a lot of the life processes that our bodies undergo to grow and develop and simply to live day-to-day life involve chemical reactions. Right. Now, a chemical reaction is a process that changes some reactant molecule that you start with into a product molecule that you end with. There can be very slow reactions and also very quick reactions. An example of a slow reaction is how iron rusts, and a very fast reaction might be when you strike a match and it lights on fire. Now, in chemical reactions, we are always going to be breaking bonds in our reactant molecules, and then we're going to be forming bonds to create our product molecules. Remember, bonds hold atoms together within a molecule. So, for example, this right here is a methane molecule. It has the formula CH4. So this is a carbon, and these are 4 hydrogen atoms. And if we put methane in the presence of oxygen gas, and oxygen gas is O2, just 2 oxygen molecules that come together. If we join those 2 things together, a chemical reaction occurs in which we produce carbon dioxide, carbon and 2 oxygens, and 2 water molecules. So in this example, our methane molecule and oxygen molecules are our reactants, what we start with, and our carbon dioxide and our water molecules are our products, or what we end with. Notice that we have to break the bonds between the carbon and the hydrogens, and we also have to break the bonds between the oxygens in order to create these product molecules. And we're forming new bonds here between carbon and oxygen, and here between oxygen and hydrogen. Now, in a chemical reaction, some reactions that occur are going to overall release energy. Some of them are overall going to absorb energy when going from your reactants to your products. So let's take a look at these graphs down here. These are showing us the energy changes that occur as we go from the reactants or starting molecules of a reaction to the products or end molecules of the reaction. On the left, we have an energy absorbing reaction where we have to take in energy in order to get to our product molecules, in order to form them. Here, this is an energy releasing reaction where overall we're seeing a release of energy as we go from our reactants to our products. Typically, an energy absorbing reaction is when we take smaller molecules and build larger ones with them. And an energy releasing reaction is when we have larger molecules that we then break down into smaller molecules. So an example of an energy absorbing reaction might be dehydration synthesis, because we're building a larger molecule, and an energy releasing reaction might be hydrolysis, because we're breaking a larger molecule down. Okay, notice in both of these images we have something called the activation energy. The activation energy is the energy required to start your reaction so that it proceeds from your reactants to your products. In an energy absorbing reaction, that activation energy is very, very high. We have to input a lot of energy to go from reactants to products. Notice in our energy releasing reaction, we're not always releasing energy in this pathway. In fact, in the beginning, we have to have a small input of activation energy. Once we input that energy, however, we're releasing a lot of energy to get down to our products. Okay. Now let's say you have a reaction that's really super slow and inefficient, or you have a reaction that has a ridiculously high activation energy. We're going to need a little help with those reactions so that they proceed efficiently within our bodies. So they need something called a catalyst. A catalyst is a generic term for a substance that's going to lower the activation energy of a reaction, which will speed it up because you don't have to put as much energy in to get the reaction started. And putting energy in takes time. Now, enzymes are a specific type of catalyst. They're a specific type of catalyst that are protein molecules. So I would say enzymes are protein catalysts. Now, this graph right here is showing how an enzyme affects the activation energy of a reaction. Since it's a catalyst, we're going to expect that it lowers the activation of an energy of a reaction to make that reaction go faster. So, in blue, we're seeing the normal reaction pathway when an enzyme is not present to assist with the reaction. Notice that the activation energy without the enzyme is pretty high. So you need to input a lot of energy to get your reaction started so you can go from reactants to products. Now, in red, we see the same reaction, but this time that reaction is assisted by an enzyme catalyst. So notice that our activation energy with the enzyme is a lot smaller. So if we don't have as much energy to put in, our reaction is probably going to proceed more quickly. Okay, now let's talk about the shape of an enzyme and why it's important. Typically, the shape of a molecule determines its function. Enzymes always have a specific portion of that enzyme or a site or location where you bring in your reactants and they are in a good location to join together, or in some cases you're taking one reactant and breaking it apart. Now, the actual site or location where enzymes bring those reactants together is called its active site. So here in blue, we have an enzyme, and this little squiggly part is the active site. Notice we also have this purple molecule, which is called a substrate. A substrate is another name for a reactant molecule that's involved in a reaction assisted by an enzyme. So a substrate is really just a reactant, but it's a specific name for a reactant when it is being assisted by an enzyme. Okay, so that substrate is going to come into the active site, and you notice that the shape of the substrate and the shape of the active site are very similar. So this enzyme can really only fit with this substrate and assist with a particular reaction. So enzymes are very specific to the reactions that they assist with. Now the substrate, or the reactant, has to fit exactly into the active site, like we saw with this picture right here. So we often call the interaction between an enzyme and its substrate a lock and key interaction, or we call it the lock and key model. So let's say this key right here is our enzyme, and this is the lock. The lock is going to represent our enzyme. The key is going to represent the substrate or reactant molecule. The key is going to fit into a specific part of the lock, the keyhole, and that keyhole represents the active site of the enzyme. And it has to be a very specific fit between the key or substrate and the keyhole or active site. Okay. Once the enzyme has assisted with the reaction within its active site, it's going to release the products of the reaction. Now, in this example right here, we're seeing a single substrate entering an active site, and the enzyme is going to help to break that substrate into two pieces. And those two pieces are the products of our reaction, and they get released. Notice the enzyme isn't really affected at all by the reaction, so it can be reused for the same job many, many times. So this is an example of where we have a larger substrate that's broken down into smaller products. However, we also can have more than one substrate, like in this example, entering the active site and being joined together by the enzyme to create a larger product. So an enzyme can join substrates together or break substrates apart. Okay. Certain enzymes work best at certain pH values and also certain temperatures. The enzymes within your body are going to function best around a temperature of 98.6 degrees. Let's say you had a really high fever. That could affect the enzymes in your body because it causes them to lose their shape. And if they don't have that particular shape, they're not going to fit their substrate like a lock and key, and they're not going to be able to do their job. So at really high temperatures, enzymes lose their shape. we call that denaturing. So we say that enzymes denature at a high temperature. Also, enzymes in your body require a certain pH in order to function appropriately. If the pH is drastically higher than it should be or drastically lower than it should be, your enzyme will also denature or lose its shape and be unable to do its job. Different enzymes in the body function best at different pH values. For example, enzymes that are active in your stomach function really well at very acidic pHs like 1 or 2. Enzymes in your small intestine, which is actually joined to your stomach, function best at a more basic pH of around 8. So depending on the location in the body, you might have enzymes functioning at vastly different pH levels. Most of them, however, are going to do best at a pH of around 7 since your body is mostly water. But something like the stomach is very specialized to help you digest your food by secreting acid, so the enzymes in your stomach have to work best at a lower pH. Now, we can summarize the role of enzymes in a couple ways. The first one is kind of what we've been talking about, regulating chemical pathways or chemical reactions by helping them to speed up. Also helping us make materials by joining substrates to make larger products. Releasing energy as well. So if you're breaking a large substrate down into smaller products, usually that reaction releases energy overall. And then also some enzymes are involved in transferring information. We use enzymes to make copies of our DNA when we're creating new cells, since new cells need to have copies of all the DNA. And we also use it to create another nucleic acid called RNA, which can be used to send information in your body. Okay. And that's it for enzymes. All right. Hope this was helpful. Thank you.